Our first fossil evidence of life comes about 3.5 billion years ago. And for almost 2 billion years, there's no evidence of any other cell type except for those prokaryotes. But then about 1.8 billion years ago, we have evidence of eukaryotic cells. So the exact manner of the origin of eukaryotic cells is still being debated. But in modern eukaryotes, we have DNA in the nucleus. We have a variety of membrane-bound organelles. We have a variety of membranes that are made out of the plasma membrane and are interconnected sets of organelles made out of membranes. And then we have the mitochondria in all of our eukaryotic cells and chloroplasts in those that perform photosynthesis. How all of this actually came about is a little fuzzy. There is evidence of both bacteria and archaea DNA in the genomes of eukaryotic cells. And some of this is probably direct vertical evolution. Some of it is possibly and probably is horizontal gene transfer as well. And then our mitochondria and our chloroplasts have their own DNA that is different from the genomic DNA. And these two organisms come about by something called endosymbiosis. Endosymbiosis is where one organism lives inside the other. So the endosymbiont forms a relationship that is inside the other one. Endo means inside, symbiont means a mutually beneficial relationship. And both the host and the endosymbiont become dependent upon one another. So the gist of this theory, the endosymbiont theory, is that a larger cell, likely already with a nucleus, engulfed a smaller cell and they became dependent on one another. Now, how they came about and became dependent on one another is a little bit of speculation. They could have been two organisms that were in close proximity and sort of started to depend on one another. But whatever happened, one ended up inside the other, it wasn't digested, and now the eukaryotic cell, cannot be separated from the mitochondria or the chloroplast or both, depending on the cell type. So the mitochondria and the chloroplast both have their own genomes, and this is different genomic DNA than the nucleus. The mitochondria and the chloroplasts do still depend on the cell. They're not independent from the cell. There are nuclear genes that are going to interact with the mitochondria, but all of the proteins that the mitochondria and the chloroplast need inside are going to come from their DNA. The mitochondria likely came from the modern group of bacteria called proteobacteria. Proteo means early or first, something like that. So this is just a kingdom of bacteria, and this is based on molecular evidence. The DNA sequences in the mitochondria are similar to this group of bacteria. So the endosymbiont theory says that an ancient archaea, because our nuclear DNA is similar to archaeal DNA, evolved the ability to invaginate or pinch in its membrane, and that membrane eventually formed an envelope around the DNA. And then from there, the endomembrane system would have evolved. So your rough ER, your Golgi apparatus, your vesicles, etc. Then this archaea cell that already has this membrane-bound DNA would have engulfed this proteobacteria. And the proteobacteria is important because that proteobacteria could use oxygen that those early cyanobacteria were producing and use it to turn cellular energy into ATP energy. So this was great because it would have provided energy to the cell. This bacteria would have got sugars, it would have got nutrients, it would have got protection from this ancient archaea. And then in turn, of course, it would convert all that lovely carbon dioxide that's floating around, all of that oxygen that's floating around as byproduct of photosynthesis into ATP. So all of our eukaryotic cells are going to have the archaea nuclear DNA, and then the mitochondria. In some in some cell lines, specifically the algae and plant lines, they are going to also have chloroplasts. So this is where a second endosymbiotic relationship took place, where the cell that already engulfed a mitochondria, the cell line, then engulfed a second bacteria. And in this second endosymbiosis event, that early eukaryotic cell that already has the nucleus and the mitochondria would have pulled in a cyanobacterium, because remember, those are the bacteria group that can perform photosynthesis. So in this event, we get the evolution of a plastid. A plastid is a general group of organelles that have evolved to store pigments. Now, we are most familiar with chloroplasts, but chloroplasts are not the only type of pigment that are found in cells. So these plastids generally perform photosynthesis of some type. They might have a specialized type of pigment that is a different color. There are other pigments, other types of plasmids that have some other slightly different functions in plants. But we are most worried about the chloroplasts because we're going to continue on our journey and talk about a little bit more about cyanobacteria, but also about algae and the plant lineage. So this diagram shows the endosymbiont theory, the origin of the eukaryote cells. So this shows the ancient archaeon with its nucleoid region. Remember that bacteria have a nucleoid region. The archaea would have had DNA in a similar region. And this archaeon evolves the ability to invaginate this membrane. And you can see over time it would have surrounded the nuclear envelope. It is in current eukaryotic cells, the plasma membrane is the nuclear membrane is derived from the plasma membrane. On the right-hand side, we see the engulfing of the ancient bacteria. Now, there are two things about this theory that indicate to us that the mitochondria and the chloroplasts were originally separate cells. One is the fact that they have their own nuclear DNA, and that DNA would have evolved quite a bit. They lost a lot of it because they were now dependent on the larger cell, so they didn't need some of their own genes. The other bit of evidence is that the mitochondria and the chloroplasts both have double membranes. Now, the first membrane is their own membrane that originated from that bacterial cell. The second membrane originates from this membrane that is surrounding it as it is engulfed, as it is pulled in by phagocytosis, and that plasma membrane wraps around the smaller cell. And so now, if we observe these organelles, they have the remnants of both of these membranes. They have an inner membrane that would have been evolved from the bacterial membrane, and then an outer membrane that would have evolved from the plasma membrane. So this lineage that only brings in the mitochondria is going to go on and evolve into our animals, fungi, and some of our protists. Some of those cells, or at least one lineage, then would have gone on its merry evolutionary way and done this again, engulfed an ancient cyanobacterium, and that line of cells that engulfed the cyanobacteria evolved into the algae line and then into the plant line. Very, very quickly after we see eukaryotes in the fossil record about 1.8 billion years ago, we begin to see multicellular organisms. So our oldest multicellular organisms resemble modern red algae and a variety of other types of algae. This picture here is something called a volvox. It is a photosynthetic, simple-celled multicellular organism. Excuse me. We'll talk about that in just a second. But there are two possible origins for multicellularity. As we move into plants and animals, we'll see that multicellularity actually evolved a couple of times. It evolved in the line that is going to lead to animals and fungi, and then it also evolved in the line that's going to lead to plants. So it is possible that there were many individual cells that were associated with one another, and they formed a colony. And from there, that colony evolved into an interdependent group of cells that then diversified into different cell types. The other possibility is that a single cell divides but stays stuck together. Now, some bacteria do this, but in this step, essentially the same thing would happen. You would eventually get a colony type structure, and the cells in the colony that all were produced from division would have evolved into different cell types. So an example of multicellularity is seen in some current organisms. These are called the volvocine algae. This image on the right is the same as the image on the previous page. And within this group, there are a variety of different levels of multicellularity in their organism. So if we looked at this first one with 2 flagella, it looks very similar to the second one that is a set of 8 cells that are associated with one another. Now, this is considered a multicellular organism, but you can see we've got those cells and we've got the flagella. Next, we have slightly larger group of organisms that are very, very similar, but also have 2 distinctive cell types. They have somatic and they have reproductive. So our somatic cells are our non-reproductive. These are the ones that are basically doing the pulling in the food and processing and interacting with the environment and all that. And then we've got this Volvox aureus. This is much, much larger organism, much more complicated, except it still only has those 2 cell types, or I'm sorry, it does have those 2 cell types. It's got somatic cells and reproductive cells. And so we can see kind of how multiple cells coming together. If we look at this as steps in an evolutionary pathway, we can see how we can get multicellular organisms from a single one coming together. And the same idea would make sense for a single one dividing and sticking together, forming a multicellular organism through the process of natural selection. So multicellularity demonstrates the idea of emergent properties. And emergent properties say that as you organize smaller levels, as you organize something into more and more complex levels of organization, they take on new functions. And so as you get more cells together, you can begin to get specialized cell types within an organism, which is going to be very, very efficient for the cell or for the organism altogether, because you can very quickly specialize. Just like we have specialized organelles in our cell, you get specialized tissues in animals and plants and fungi as well. And that's going to make doing the jobs in the organism much more efficient. So as our multicellular organisms become more common, there are a few common characteristics of these multicellular organisms. So multicellular organisms start out as a single cell, and then they divide. So if you think about a sperm fertilizing an egg, you have one cell and then it divides and divides and divides, and eventually it grows into a multicellular fetus, animal, child, whatever direction you want to go with it, and eventually grows into a full-grown multicellular organism whose cells are all adhering to one another. So that parent cell, where the sperm and the egg has fused, begins to divide, and those divided cells are called the daughter cells. Those daughter cells then begin to differentiate. Differentiation means they all take on different characteristics and different functions. And those differentiated cells become different cell types within the organism. So they might become muscle tissue, they might become connective tissue, they might become epithelial tissue, which is all the tissue that lines the insides of our body. They might become neurons. And so they all have these different fates that do a particular job within the cell. As organisms get larger and larger and larger, of course, they are made up of more and more cells. Most of those cells are going to be somatic cells. These are the cells that carry out all the normal activities. Those that are not somatic cells are the reproductive cells. So in large animals, we have very small number, relatively speaking, of reproductive cells, and their goal is just reproduction. All of the other cells are there for survival of the organism. So we're going to go through all of our major organismal groups, starting first with our bacteria and archaea, and even some viruses. And then we're going to get into the characteristics of our multicellular groups, our protists, eukaryotic groups, protists, and then our multicellular groups, fungi, plants, and animals. And so we're going to talk about the major characteristics that distinguish each of these groups. There's some examples of the characteristics we're going to talk about for animals. You don't need to know all of these yet. We'll get to them a little bit later. But we're going to go through our simple multicellular animals, our invertebrates. We're going to start with sponges and our jellyfish, and then we're going to move into more complex structures, those with bilateral symmetry, or where symmetry is on the same on two sides, but different front to back. We're going to talk about how they move. We're going to talk about the different ends, the anterior, posterior end. So all of these are things we're going to talk about with animals. We're going to talk about similar trends in plants and fungi.